Anjali Karki, welcome to New Mexico in Focus. Thanks so much for being here.
Thank you for inviting me.
So, before we get into the new mosquito food that you've developed, let's talk about the need for such a thing. Why are large populations of mosquitoes being reared in labs across the world?
It almost sounds like backward, right? Mosquitoes are the dangerous animal known to humans, and funnily enough, we are actually rearing large number of mosquitoes in the research facilities. So mainly, these mosquitoes are reared for the research purposes and for the population control strategy.
What kind of research?
So there are different types of purposes, and it could be for the new insecticide resistance, or it could be for the repellency testing, it could be for the antiviral drug development, or for the mosquito virus interaction. It could be also for the vector population control, and so on.
Okay. One method that lab mosquitoes can be used for is something called the sterile insect technique. This is something that could be used rather than, say, insecticides to kill off mosquitoes to prevent the illnesses that they're vectors for. Can you talk about how that works?
Of course, this is a really interesting technique that has been developed way before my time. So the way sterile insect technique works is it is an eco-friendly form of the population control methods, and it involves mass rearing of the insects. The way it works for the mosquito is that male mosquitoes are screened from the female mosquitoes, and then they are sterilized, and these sterile males are then released into the wild, and then these males will compete with the wild males and then mate with the wild female mosquitoes, which will render these female mosquitoes to lay unfertilized eggs, which will never hatch. And by this way, they are expected to control or reduce the population in the next generation.
Okay.
Uh,
With the prospect of releasing millions of these mosquitoes into the wild, I'd imagine comes with some trepidation or fear. Where is this being done and how successful has it been?
so it has been done in the field like the control of the New World screwworms. It has been also done in fruit flies and for TT flies. And it has been done for over 70 years. But the success rate, that is something that requires the regular monitoring, surveillance, and then a regular field release and all. So it depends on how well the investigation has been going on and all.
Okay. Well, in order to raise these sterile mosquitoes in the lab, they have to eat, right? They have to get nourished. How is that typically done?
So the common understanding is that all mosquitoes are bad or they bite humans. But the thing is, only female mosquitoes bite humans, and this is because they require the nutrients from the blood to lay eggs. So male mosquitoes can survive on the nectars or the fruit juices and sugar solutions, which is provided in the laboratory setting. So at present, the mosquitoes are provided this sugar solution and then the blood meals in the lab either by using the live animals or the isolated vertebrate blood from the animals.
Okay, so isolated blood is fed to, it sounds like just the female mosquitoes. Is that right?
Yes.
And that is from live animals. How is that sourced?
That is usually that is something provided commercially, like us from the slaughterhouse or the blood banks, or either by directly using the live animals. Um, it is a very complex process, I guess. I don't know the details about the how it is manufactured and bottled and provided to the different facilities, but it involves different testings and use of different anticoagulants and all. Um, but it is a very expensive process um, and requires a trained personnel to handle all the things, and it could be expensive.
So the expense could be a drawback to using this method. What are some of the other drawbacks? What were some of the problems that your research is trying to solve for?
So a problem that we often see with the mass rearing of the mosquitoes in the laboratory setting is the need of the continuous supply of the fresh vertebrate blood. which could be expensive. Not only the expense, but the way it is handled. It needs to be refrigerated properly, and that the personnel which is handling the blood during the blood feeding needs to be trained. So those are all the things that are the reason that makes the demand for the alternatives to these blood meals interesting.
Are there any? I mean, some of this research is happening worldwide. I'd imagine there's some ethical concerns for particular cultures around using live animal blood.
Yes, there is some ethical use of the live animals has its one ethical, economic, and safety concerns associated with them, and they have different kind of complex logistic that is involved in them, which can make the laboratory work push back the laboratory work to pass through those ethical concerns and ethical reports and all.
Well, so your team at NMSU has developed an alternative to using isolated blood to feed these mosquitoes, which you call Skeeto Snack. I love the name. What is the alternative made of?
Thank you for the introducing the diet. That name is really interesting, and that is something my professor, Dr. Imo Hansen, come up with. So this diet is made of all the ingredients that could be retained from the actual blood. So it has bovine serum albumin, which is a source of amino acids. It has iron source. It has cholesterol source. It has different salts and buffers, which mimics the pH of the blood. And it has also phagostimulant. So phagostimulant is something that stimulates the feeding behavior in insects. So it is a complex mixture, or it is a detailed mixture of all those nutrients, which is in an optimized form to provide or to maximize the egg number, hatch rate, and all for the mosquito rearing.
Okay, and it resembles blood in those ways. Is it synthetic, or where are you sourcing the ingredients?
All of the ingredients are commercially sourced or commercially available plant animal source, I would say.
And what has been the process in your lab for kind of perfecting the recipe, finding out what the mosquitoes actually like?
So this diet is actually the result of the hard labor of many other former graduate students in the lab. But for this research, the strategy that I have used to develop this diet is that I modified the mosquito snack recipe either by adding or removing one thing at a time. So thereby I have prepared several different versions of this diet. And then I did an extensive series of feeding assay where the optimized mixture of the nutrients for the Anopheles mosquitoes was developed to maximize their egg or feeding rate and then larval hatching. And I also screened for the iron sources, like the phagostimulant source, and then the nutrient source
And
and the buffer.
I was reading in your paper that the mosquitoes actually appear to like Skeeto Snack better than blood.
That is correct.
How can you tell?
If we can tell that by doing a choice assay, in order to figure out what is the preference of these single mosquitoes whenever they are given a choice between the Skeeto Snack diet and then the blood. So this is something that is done in collaboration with Dr. Peter Antonio at Texas A&M. And this is a really fun collaborative work, and it is present in my paper also. So we give individual mosquito a choice between a Skeeto Snack diet and a blood, and they show preference for the Skeeto Snack diet. And we have actual data to show that.
Well, good work on that. The original Skeeto Snack was developed for mosquitoes that are responsible for transmitting yellow fever, dengue, Zika. Did you have to update the Skeeto Snack for the mosquito that carries malaria? Or is it just that you had access to a mosquito that carries malaria in order to test this out on?
The thing is, we actually optimize this diet or for the malaria mosquitoes. So yes, just like you said previously, in 2018, this diet has been developed for the yellow fever mosquito Aedes aegypti, but our preliminary study shows that Anopheles mosquitoes, which are a vector for malaria, do not like to feed on this diet. And that's where my research comes in.
Just last month, mosquitoes that carry West Nile were detected here in Bernalillo County along the Rio Grande. Could theoretically Mosquito Snack be used to nourish those types of mosquitoes in the lab if a sterilization technique were to be used?
I really like this question because this diet is supposed to be, in a bigger picture, it is supposed to replace the blood, which is required for the reproduction in mosquitoes. And Culex mosquitoes, which are actually the vector for this West Nile viruses, also requires the blood to reproduce or to lay eggs. So we expect this mosquito snack diet to work on West Nile virus transmitting mosquitoes also.
Also, recently, the New World screwworm, which is a flesh-eating parasitic fly, cropped up last month in Texas. One case in New Mexico in a dog. Same deal. It could potentially be used for that fly.
Just like you said, it is a parasitic fly, and then the real danger comes from the larvae, which actually eats the flesh of the animals. Um, it matters whether the blood has a role to play in its reproductive cycle for the New World screwworm, which turns out that the adults of the New World screwworm doesn't feed on blood, and there is a differences in their anatomy and their reproductive cycles between the mosquito and then the New World screwworm. So for the mosquito snack to work on New World screwworm, it would definitely require some modification, I would say, for the mastering. But as it is, I couldn't say anything without the proper or rigorous testing.
Okay. Now, have you distributed the Mosquito Snack at this point, or where do things stand with this actually getting to be used in labs across the world?
That is the bigger theme for this whole research. My PI, Dr. Imo Hansen, has been working tirelessly on this development of artificial diet for over a year, or over since 2015. And at present, we have this interesting opportunity to collaborate with IAEA, which is the International Atomic Agency, Atomic Energy Agency. And so as we speak, it is in the R&D process right now. So hopefully in the future, we would be able to distribute or market it as a artificial diet for mosquito rearing throughout the globe. But free of cost, we have distributed this diet to different labs within the US. But it is just for testing because they reach out to us saying that, hey, can we test this diet for our mosquito or laboratory mosquitoes? And we just provided them, but not in a bigger scale.
All right. Well, congratulations, Anjali, on your research. And thanks so much for coming on New Mexico in Focus to explain it to us.
Thank you so much for having me.